السلام عليكم يا شباب. صبحكم الله بالخير. Today I'll start talking about the histology of the digestive system. The digestive system is one of the important systems in our body. It breaks down food stuff into small molecules that can be easily absorbed. For example, ingested proteins are first degraded into polypeptide. Then they further broken down into small peptides and amino acids, therefore can be absorbed. The digestive process starts in the oral cavity where ingestion, fragmentation, and mouthing of food occur. The oral cavity is also involved in speech, facial expression, sensory perception, and breathing. In this lecture, I will talk about the oral cavity. Additionally, I will introduce the general plan of the GI tract. Inshallah, at the end of this lecture, we should recognize and identify the parts of the digestive system. We should be able to describe the normal structure of the oral mucosa. We should describe the normal structure of the tongue and enumerate the names of salivary glands. We should identify and describe the normal structure of the four layers of the wall of the GI tract. And finally, we should recognize the structure of the esophagus. The digestive system consists of the alimentary canal, which starts at the oral cavity, going through the esophagus, stomach, small and large intestine, and down to the anal canal. The structure in this system secretes enzymes and hormones. These function in digestion, absorption of nutrients, and elimination of indigestible material. Additionally, the inner layer of the GI tract, the mucosa, perform an important barrier function. They separate the content of the luminal content of the GI tract from the internal environment of our body. The oral cavity is formed of the vestibule and the oral cavity proper. The vestibule lies between your teeth and your cheeks, this area. Behind your teeth, you will find the oral cavity proper. It is bounded superiorly by the hard palate and soft palate. Inferiorly, it is bounded by the tongue and the floor of the mouth, and it opens posterior to the oropharynx. The oral cavity is lined by the oral mucosa. This mucosa is formed of epithelium and underlying connective tissue, which is a dense, irregular, collagenous connective tissue. In most parts, the epithelium is stratified squamous epithelium. The oral mucosa has three types. The first type is the lining mucosa, and this type can be found in the soft palate, lips, cheeks, floor of the mouth, inferior surface of your tongue, and alveolar mucosal surface. At these sites, the lining mucosa covers muscles like your lips, cheeks, and tongue, bone as an alveolar mucosal surface, or glands as in soft palate, cheeks, and inferior surface of your tongue. The epithelium of this mucosa is non-keratinized. The second type is the masticatory mucosa, and this type can be found in the gingiva and hard palate. The epithelium of this type is keratinized. The last type is the specialized mucosa, and this type is associated with the taste sensation. This type is restricted to the dorsum of your tongue. This part has papillae and taste bud for generation of the chemical sensation of taste. The lips guard the entrance into your oral cavity and digestive system. It has three surfaces. The outer surface covered by thin skin, inner surface lined by oral mucosa, and a transitional zone between the two surfaces called red margin or vermilion border. The thin skin of the outer surface is heavily innervated by sensory nerve. Like thin skin and other areas of our body, it is formed of epidermis, which is a stratified squamous epithelium keratinized. Underneath the epithelium, there is a dermis with hair follicle, sweat gland, and sebaceous gland. The red margin is also covered by stratified squamous epithelium. This epithelium is lightly keratinized. This border appears red or pinkish in color because the epithelium is transparent to the blood in the capillaries underlying connective tissue. This border lacks hair follicle. It has no gland, therefore it is dry, and it is kept most by saliva from your tongue. The inner surface of the lips is lined by thick, non-keratinized stratified squamous epithelium. Underneath the epithelium, there are aggregates of minor salivary glands. The core of the lips is formed of dense, irregular connective tissue. This tissue contains a bundle of skeletal muscle fibers. These fibers allow the lips to be highly mobile for ingestion, speech, and other forms of communication. The tongue is a highly muscular organ. It protrudes upward and forward into the oral cavity from the floor of the mouth. It has two surfaces. The upper surface or dorsal surface. The lower surface we can see here, it's called the ventral surface. The ventral surface of the tongue is covered by stratified squamous epithelium. This epithelium is continuous with the epithelium of the floor of the mouth. The dorsum of the tongue is covered by stratified squamous epithelium keratinized. Underneath the epithelium, there is a connective tissue which penetrates between the bundles of skeletal muscle fibers. The mucous membrane, which is formed of the epithelium and the underlying connective tissue, is strongly adherent to the muscle core. The dorsum of the tongue has a V-shaped groove called sulcus terminalis. The sulcus terminalis points posteriorly. It divides the dorsum of the tongue into anterior 2/3 and posterior 1/3. Let's zoom here a little bit. Here is the sulcus terminalis. This is a part of the anterior 2/3, and this is a part of the posterior 1/3. In the anterior 2/3, there is irregularities and projections. These projections correspond to the lingual papillae. Similarly, the posterior 1/3 show bulges. These bulge due to the presence of the lingual tonsil and a small aggregation of lymphoid nodules. There are 4 types of lingual papillae. The first type is the filiform papillae. These are the most numerous papillae, and they are scattered all over the dorsum of the tongue. They are tall and narrow pointed papillae and covered by highly keratinized stratified squamous epithelium. This gives them gray or whitish appearance, like this little dot in the dorsum of the tongue here. They provide rough surface for movement of food during chewing. These papillae lack taste buds. The second type of lingual papillae is the fungiform papillae. From their name, fungi, they are mushroom-shaped structure. Their epithelium is poorly keratinized or non-keratinized. They have a highly vascular connective tissue core. This makes them appear like red spots in the dorsum of the tongue. They have taste buds in the covering epithelium. Those at the tip of the tongue detect a sweet taste, while those somewhat away and towards the side of the tongue detect salty taste. The third type of lingual papillae is the foliate papillae. They present in the edge of the tongue as a parallel ridge in front of the sulcus terminalis. They have taste buds in the facing wall of neighboring papillae. Serous glands open into the cleft between the neighboring papillae. They are poorly developed in adults, however, they can be recognized in younger individuals. In some animals like rabbit, these papillae are well developed and constitute a great aggregates of taste buds. The last type of lingual papillae is the circumvallate papillae, also called vallate papillae. There are 10 to 14 vallate papillae. They are arranged as a single row just in front of the sulcus terminalis. These are large dome-shaped structures surrounded by a depression called moat. The moat is lined by stratified squamous epithelium containing taste buds. Minor salivary gland called von Ebner's gland open into the base of this moat. They clean the moat to enable the taste buds to rapidly respond to the changing of stimuli. Taste buds are the structures that detect taste in our food and beverages. There are 250 taste buds in the lateral aspect of the vallate papillae, with many others in the fungiform and foliate papillae. Remember, filiform papillae do not have taste buds. Taste buds appear as oval, bear-staining structures that extend much of the thickness of the epithelium. Underneath the taste buds, there are nerve fibers that are also lightly stained. Let's zoom here a little bit. The apical part of the taste buds contains a depression. This depression is called taste bore. 3 types of cells can be recognized in the taste buds. The gustatory or neuroepithelial sensory cells, they project their microvilli into the taste bore. Their bases synapse with sensory nerve axons, which perforate the basal laminae of the taste buds. In addition to the gustatory cells, there are supporting cells whose function is not well understood, and basal cells for generating the other types of cells. Taste and dissolved in saliva, make contact with the microvilli and interact with cell surface taste receptors. I have just finished talking about the anterior two-thirds of the dorsum of the tongue. Now, I'm going to talk in brief about the posterior third of the dorsum of the tongue. This part is covered by a relatively smooth stratified squamous epithelium. The epithelium overlies a lymphoid follicle of a lymphoid tissue called lingual tonsils. Lingual tonsils, together with palatine tonsils and adenoids, form the Waldeyer's ring. This ring guards the entrance to the GI tract and the respiratory system. Like palatine tonsils, epithelial crypts penetrate into the lingual tonsils. Salivary glands are exocrine glands found in our oral cavity. They produce saliva which has digestive, lubricating, and protective functions. We have minor salivary glands and major salivary glands. Major salivary glands include parotid gland, submandibular glands, and sublingual glands. These glands are surrounded by a dense connective tissue capsule. The capsule sends septi or trabeculae into the glandular substance. These septi divide the gland into lobes and lobules. A neurovascular bundle runs through the trabeculae to supply the gland parenchyma. We have three types of secretory units. We have serous acini and mucus acini, this part, and seromucous acini, which is composed of mucus part covered by a moon-shaped serous cells called serous demilion. The acinar cells rest on basal lamina. Between the basal lamina and the acinar cells, we have myoepithelial cells. These cells form networks around the secretory units, while they take more longitudinal orientation along the duct system. Myoepithelial cells contain actin and mucin filament, and their contraction propels the secretion out of the secretory units into the duct system. We have the serous acini. The serous acini secrete proteins that are non-glycosylated, such as digestive enzymes. The acinar cells are pyramidal in shape, and they have rounded vesicular nuclei and basally located. They are located towards the base of the acinar cells. The apical part of these cells contains zymogen or secretory granules. If we examine these cells with the electron microscope, we will recognize that they have the structure of protein secreting cells. They have abundant rough endoplasmic reticulum and Golgi complexes. In addition, they have secretory granules in various stages of maturation. These secretory granules store secretory products which are enzymes until they are released. Serous acini secrete watery fluid, which include water, electrolytes, and enzyme like salivary amylase and lipase. These enzymes start the digestion in our oral cavity. Unlike serous acini, mucus acini produce heavily glycosylated proteins. Mucous acinar cells have small flattened nuclei located at the basal edge of the cells. They have flat, basally located nuclei. Their cytoplasm is lightly stained. This is because the secretory granules in this cytoplasm fail to stain with the common histological stain. If you examine these cells with electron microscope, they have rough endoplasmic reticulum and Golgi complex. Unlike serous acinar cells, which have electron dense granules, Mucous acinar cells have electron-lucent granules in their apices. These acini, the mucous acini, produce mucin, which become hydrated upon releasing from the gland and to form mucus. The last type of secretory units is the mixed acini. Sometimes they are called seromucous acini. From their names, they have a mucus secretory portions and serous secretory portion. The serous secretory portions cap or form a moon-shaped structure on top of the mucus secretory portions. This cap of serous cells called serous demyelin. Serous demyelins. These acini produce digestive enzymes and watery mucus. These secretory units are drained by the salivary gland duct system. This system starts with intercalated duct. These ducts are lined by simple cuboidal epithelial cells. Saliva in the intercalated ducts is isotonic and is called primary saliva. Intercalated ducts give rise to striated ducts. Striated ducts are lined with simple columnar epithelial cells. These cells have basal striation. There are striations towards their basal parts. If you look to one of these cells by the electron microscope, you will find mitochondrial rich basal enfoldings. At these enfoldings, sodium ions are pumped out of the cell, while potassium and bicarb pumped into the cells. And finally, they are released into the lumen of the striated ducts. By now, the striated ductal lumen contains hypotonic saliva, and it's also called secondary saliva. Striated ducts give rise to interlobular ducts, which are lined by stratified epithelium. These further unite to form larger excretory ducts. In the stroma around the acinar cells, there are plasma cells. These cells secrete immunoglobulin A. Immunoglobulin A is taken by the acinar cells and the striated duct cells. In these cells, the secretory component is added and is to form the secretory immunoglobulin A, which released into the lumens of the acini and the striated duct. Here we have the major salivary glands. Let's start with the parotid gland. It has a prominent connective tissue capsule and trabeculae. Its secretory units are purely serous. It has prominent striated ducts with their characteristic striations. It becomes invaded by adipose tissue at the age of 40. The submandibular glands is another type of major salivary glands. They also have extensive connective tissue capsule and trabeculae. 80% of their secretory units are serous acini. The rest are of mucus types. Serous demyelins are limited in these glands. Finally, we have the sublingual gland. These glands are the smallest. 99% of secretory units are of mucus type with serous demyelin. The rest are serous acini. The wall of the gastrointestinal tract is formed of 4 layers. The innermost layer is mucosa. Then we have the submucosa muscularis externa. The outermost layer is adventitia or serosa. The mucosa includes lining epithelium. Underneath the epithelium is a layer of lamina propria. This layer is formed of loose connective tissue which contain blood vessels, lymphatics, and cells of the immune system. A layer of smooth muscle cells called muscularis mucosa separates the mucosa from the next layer, which is submucosa. The submucosa is composed of dense connective tissue, which is larger blood vessels and lymphatics. It contains the mesenteric plexus of autonomic nervous system. In certain locations, some mucosa has glands or aggregates of lymphoid tissue. We then have the muscularis externa. This layer is composed of smooth muscle cells in most parts of the GI tract. Muscularis externa has two layers. Inner circular layer. In this layer, the smooth muscle cells are helically or circularly arranged around the lumen. Outer to this layer is outer longitudinal layer. In this layer, the smooth muscle cells are arranged longitudinally. In between the two layers, the inner and outer layer, there are a thin layer of connective tissue with lymphatics, vessels, and the Auerbach or mesenteric plexus of the autonomic nervous system. Outside to the muscularis externa, we have the serosa. The serosa is composed of a thin sheet of loose connective tissue with blood vessel, lymphatics, and adipose tissue. This layer is covered by a layer of simple squamous epithelium or mesothelium. Serosa is found in the organ which covered by the peritoneum. Extraperitoneal organs like the esophagus, they have a thick layer of connective tissue called adventitia. The adventitia link the esophagus to the nearby structure. The esophagus is a muscular tube. It transports ingested food from the pharynx to the stomach. In cross section, the mucosa is thrown into folds. These folds are arranged longitudinally. They obliterate the lumen of the esophagus. The mucosa is lined by a stratified squamous non-keratinized. Underneath the epithelium is a layer of loose connective tissue with blood vessels and scattered lymphocytes. This layer is called lamina propria. Outside of the lamina propria, there is a muscularis mucosa. This layer is formed of ill-defined layer of smooth muscle cells. Muscularis mucosa allows localized movement and folding of the mucosa of the esophagus. The submucosa is formed of dense connective tissue with large blood vessels and seromucous glands called esophageal glands. Ducts of these glands transport secretions from the secretory portion towards the lumen. This secretion lubricates the lumen of the esophagus. Outside the submucosa, we have the muscularis externa with its inner circular layer and outer longitudinal layer. This oesophagus was cut longitudinally, therefore the inner circular layer muscle fibers cut transversely, while those of the outer longitudinal were cut in longitudinal directions. In the upper third of the esophagus, skeletal muscle fibers predominate the muscularis externa, while smooth muscle cells predominate the muscularis externa of the lower third of the esophagus. Now, how about the middle third? The middle third represents a transitional zone where the skeletal muscle fibers are gradually replaced by smooth muscle cells. Here in this figure of the middle third of the esophagus, unlike the previous figure, this esophageal section was cut transversely. Therefore, the muscle fibers in the inner circular layers were cut longitudinally, while those of the outer longitudinal layers were cut transversely. Back to the middle third of the esophagus. Here we have the two types of muscle fibers. Here are bundles of smooth muscle fibers together with bundles of skeletal muscle fibers in this part of the esophagus. Outside of the muscularis externa, we have the adventitia, which is formed of connective tissue that contain blood vessels and nerve fibers. The adventitia support, protects, and anchors the esophagus to other structures in the mediastinum. By now, I have just finished talking about the oral cavity, lips, and tongue. Additionally, I introduced the general plan of the wall of the GI tract. Finally, I talked a little bit about the esophagus. In the next lecture, inshallah, I will talk about the stomach.